When an animal is in its last throes of an anthrax infection, it has a lot of neurological challenge and they lose a lot of their motor function. That's usually where that animal will collapse, when they lose that motor function.
Our lab is primarily investigating bacterial outbreaks, mostly anthrax or Burkholderia or Brucella.
The history of anthrax goes way back, of course. Bacillus anthracis was the first pathogen ever characterized and described. It was also one of the first vaccines ever developed. One of our major focus in the lab is on sequencing and understanding the bioinformatics of Bacillus anthracis. And on the other side is understanding where we expect to see anthrax occurring in the environment. What are the environments where we think it'll survive?
We are working with international collaborators or locally as well in the US to investigate outbreaks and see what kinds of things are happening in that space and in that region, such as what people are living there, cultural practices, what kind of animals are living there, what's the ecology of that area. And so we compare all those together with how we study things using geography and geospatial methods and tying in all the genetic work or serological work as well to see how that outbreak or how the disease has been persisting in the area.
We want to know, how do animals come into contact with those reservoirs of Bacillus anthracis in the environment? So we do that two major ways. One is by capturing those animals, safely immobilizing them, putting a GPS collar on the animals and releasing them, and then monitoring their movements in the environment. And modeling over that same mapped environment we have for anthrax, mapping how do those animals use that environment. And then recently we've done work where we place wildlife cameras out on individual carcass sites to understand how animals come back to those carcass sites, because that forms that environmental reservoir where the animal died and contaminates the environment, sets up the next stage of infection for the next animal to come along.
Another important part is that we can train our collaborators and other peers that we have in different countries and the groups that we're working with to utilize the geospatial methods that we've been doing, as well as the laboratory wet bench methods like whole genome sequencing or serology. And we can all work together and ensure that whenever we do leave these countries, that all of the training and experience that we've brought with us is staying with them.
The lab is really this collection of collaborators that we have, and it is literally a space for collaboration.